Hi folks, and thanks for taking the time to view this presentation. I put why up here because I'd like you to understand why we're doing this, and I'd like you to understand why we do some of the things that we do when we're trying to prevent Legionella from growing. So the why we're doing it is because I have always found it easier to understand what I was doing and what I could do and what I couldn't do if I understood the reason that we were doing something. So the reason that we're making these presentations is because we want everybody to be safe. We want our staff to be safe. And that means everybody. It means you, me, and the people we work with to try to make sure that these systems across campus are functioning the way they're supposed to be and that they're not posing a threat to any of us. We also want to keep the Wayne State community safe. We have a lot of people on campus. We don't want any of them to get infected with Legionella, and that really is our obligation to do everything we can to keep that from happening. It turns out to be a little more complicated than we like, but the fact is the techniques for doing that do exist, and we just have to make sure we follow them. And then the third item for safety is making sure that we protect our resources. So we all know that a lot of this equipment we're working with costs a lot of money, and you certainly can appreciate how much money is being spent by the university on making sure that all of us are safe and that these systems that we're working on don't pose a threat to the members of our community. As for why I'm taking the time to tell you all this stuff, it's because I always hated it when people told me, well, just do it. Don't worry about why you're doing it. I can understand why sometimes people say that, but I never liked it. And so I've always tried to take the time to explain why we're doing stuff. So I thought I'd take a few minutes here to share some of that with you. As some of you know, my background is microbiology. I was trained as a microbiology, and that's what my degrees are in. So let me just walk through a few basic principles that are behind some of the stuff we're doing to control Legionella. So what is Legionella? Well, it's a bacterium. It's one of the simpler forms of life on the planet. They're single-celled. And they grow by dividing. They split in two and then keep doing that. So you can see here in this slide that they're not very big. This is 2 micrometers right here. I think if you look, you can see some of the cells are in the process of dividing. Some of them will have kind of little dark areas where the cells have recently divided. So these are small, and like David and Goliath, sometimes, small things can make a huge difference. And so we have to really keep an eye on these. And I want to emphasize that these things are not big. You can't see them. You can only see them when they're present in large groups, which I'm going to talk about in a minute. So 2 micrometers or 2 microns. And how big is that really? Well, so here's how big it is. This is sort of lets you compare to some common things that we encounter. And then right here is the bacteria at the very bottom. So, you can't see individual cells. You can't even see several cells. But you can see a whole bunch of them when they're piled on top of each other. And that's what we call a colony, which I'm going to get to in a minute. So the other thing that you can see is when they start growing together as a group or in communities, they can form what's called a biofilm. Now, I'm sure most of you are familiar with the term biofilm. Most of us encounter it in things like pipes and things that are in constant contact with water. And these biofilms are just large communities of microbes. It's not just bacteria. It's all kinds of different microbes on there, and from, very, very diverse communities. And they've also learned how to live around each other and actually help each other. So they kind of live synergistically. If you look at how biofilms form, here's a micrograph showing. So if you look, here's a scale at the bottom right here showing how big 10 micrometers is. And so these are tiny little things, and you can just look at that picture and you can see, look how many different kinds of organisms are there. These to me look like they might be some kind of protozoa or maybe a blue-green algae, and this looks like they're bacteria. There's some fungi in there. So you got a whole big happy community here, and they all sort of cross-feed each other and they help each other survive. And the fact that they're buried in that mucousy layer protects them from chemicals that are in the water, which is a problem we have to deal with. One of the things that's present in these biofilms is actually not a bacterium. It's a small animal that's single celled called an amoeba. You can see it here, kind of like the blob. It surrounds its food and eats it. They also can go fishing for food. They can send out little tendrils that bind to bacteria and bring them in, and one of their most common sources of food is bacteria. So as they say, they eat bacteria for breakfast. Which, you'd say, well, that's a great thing, then we want those in our biofilms, except for a minor detail. And that is that the Legionella have found a way to actually take advantage of the fact that they're eaten by amoeba. And so these amoebae, when they go and ingest a Legionella bacterium, what happens is the Legionella can actually grow inside the amoebae. And instead of being digested, which is what happens to most bacteria, what happens is they can resist those digestive forces inside the cell, and they actually take over the amoeba and use it as an incubator to grow more cells. A similar thing happens, so this is a biofilm like in a cooling tower or something like that. But also what happens is, when these things are starting to be shed by an air conditioning cooling tower, the cells spread through the air and into a person's lungs. And as they begin to grow in the lungs, we produce our own cells that are part of our immune system, and they're called macrophages, which literally means big eaters. And and normally these macrophages are cells that move like an amoeba does, actually, and they attack bacteria and such that are invading our bodies. Normally they eat them and digest them, and that keeps us from getting sick. But just like Legionella has developed techniques for resisting the amoebae out in the wild in biofilms, they also have developed systems for resisting the macrophages as part of your immune system. So instead of being eaten and digested, what happens is they actually reproduce inside the immune cells, the macrophages, and they use it as a mechanism to protect them from other defense systems, make more cells, and then spread throughout the lungs and the body. So if we look at the two processes, the first one, which happens in biofilms that are in pipes and stuff, they use this as a mechanism of reproduction. It's called amplification. In humans, our macrophages serve as a way to protect them from the other parts of our immune system, and these cells can reproduce and then be secreted out from these little incubators, which actually are normally we use to protect ourselves from invading bacteria. So these guys are pretty smart, and they're really a force to be reckoned with. When these cells are inside these amoebae, it's very hard sometimes to get chemicals to penetrate, and that's why treating and eliminating them is so difficult. So here you can see that, you know, the two look very similar, and they use similar mechanisms to protect themselves. So what I wanted to show you is some of the things that you're going to be doing are related to some of these characteristics that Legionella have, both in their physiology but also in their interesting life cycles. And so I took these points from this CDC website here, and I would encourage you to go look at it if you haven't already. It's not complicated, it's just got a couple of bullet points, and I'm going to go over those. But I think not only is it important to know what to do, but it's also important to know why you're doing it. So I thought I'd take a few minutes to show some of that. All right, so here's an interesting movie. These are cells, bacterial cells, dividing. And I wanted to point out that, you know, notice that it doesn't take long for one or two cells to become very many cells. And that is a key point. So one of the first points on that CDC website is to make sure that the disinfectant amounts are right by frequently biocide testing. Okay. So we, as you know, we do test frequently to make sure that the levels of biocide, which, you know, are chemicals that inhibit Legionella and other bacteria, from growing in the cooling towers. So if that amount drops below the amount that's needed to keep these Legionella from growing or to kill them, then what happens is what you see in this little video here. These cells start dividing, and they divide pretty quickly. So they can go from just a few to several thousand in just a few hours. So what does that mean to you? Well, it means that if you go up and you test one of our cooling towers and you find out that the amount of biocide is far below what it's supposed to be, that this process may already be happening. And if it happens to be on a Saturday, you may not be able to say, well, I'll just wait till Monday and then we'll take care of it. Because by the time Monday rolls around, these things may already have happened. These things may already have reproduced to where we have very high numbers, and those numbers are then being shed into the air in microscopic droplets, which then are going out and spreading around to the population. That is one of the concerns that we have. So speaking of microscopic droplets, these bacteria travel in water droplets. They really don't get around as individual bacteria. So when these cooling towers, or the evaporators are functioning, we're putting water vapor into the air, and these droplets contain Legionella. Maybe if there's Legionella in the water, the droplets will contain Legionella. And depending on how humid it is and how hot it is, these droplets can travel quite a ways. So why would humidity make a big difference? Well, as you know, when it's really humid, things don't evaporate very quickly, and those droplets can continue without evaporating for quite a while. And depending on the wind, they can travel, miles. So somebody, 2 miles from a cooling tower could inhale a droplet of water from one of our cooling towers that contains Legionella, and they could get Legionella. And so it's important that we keep an eye on these things, because it's not just what's happening immediately around the building. It can happen a long way away from the buildings, and there are many documented cases of that. So, how do we count? How do we determine if we've got enough to inhibit the growth of these organisms? Well, we just talked about one way. We can chemically test for the concentration, the amount of biocide that we have in our water. And as you know, we're doing that on a regular basis. We do it frequently because of the fact that I just told you. They can reproduce so quickly that we need to know as soon as there's a problem. If we detect low biocide levels, then we need to do something about it right away. We need to look and see what's the problem. We need to add some biocide somehow. So these are really, we have a whole team of experts that are dedicated to this kind of thing. So make sure you know what to do if you do encounter low biocide levels. What else do we do to identify when there could be a problem? Well, one of the things we do is we do dip slides. And I know you're all familiar with this. So dip slides are very simple things. They have a nutrient agar medium on them, and you, as the name implies, you dip them in the water. If the water contains bacteria and other types of microbes, those microbes will stick to the surface of the slide, and after you incubate it for a while, they'll grow colonies. And these colonies tell us how many bacteria were in the water when you stuck the slide in there. So let's take a look at how that works. So what they say is you're looking for CFUs, or colony forming units per millimeter. You know, we scientists always try to make everything as complicated as we possibly can so that nobody can understand us, and this is a good example. What is a colony forming unit? Well, as the name implies, it's something that forms a colony. So here you can see these are colonies. Each one of these dots is a colony. Typically, the theory is that a colony starts out as a single bacterium. Okay? So that when you deposit a single bacterium on the agar surface on the slide, there's nutrients in there, it starts to divide. You get 1, and then you get 2, 4, 8, 16, etc. After a while, it gets big enough to see, and they put a little dye in these things so that you can see them when they grow, and you get enough bacteria that you can actually see it, and that's called a colony forming unit, or a colony. That's a colony right there. The colony was formed by, theoretically, a single bacterium, so that means if you count the number of colonies, what you're really counting is the number of bacteria that were deposited on the slide. So why not just call them bacteria instead of colony forming units? Well, that's a good question, and I'll show you. So here is a micrograph of a growth surface. These little dark dots here are bacteria. If you look really closely, you can see that some of them, there's more than one bacterium there, and then others have a single bacterium, okay? Just a single organism, all right? So, and why is that? Well, when bacteria are growing, they divide, and then they eventually, so you have 2 of them kind of stuck together, and then they eventually break apart. Sometimes they do that and they form long chains because they stay stuck together after they divide. So you can, when they stick to the surface of the growth medium, You don't know if your colony started from a single bacterium or from 10. And there's no way to really know that. And so that's why scientists use the term colony forming unit. They assume that when you stick these slides in the water, even though some of them are going to be the result of one bacterium and others could be 10 or more, they assume it's going to be the same every time you do it. So if you repeat the process the same way every time, what you'll get may not be quantitatively perfect, but it's going to at least let you know if there's a problem or not, because you'll know if you've got 10 or 1,000 or a million. Okay, so let's look and see. Let's watch a colony get formed here. You can see that they start dividing, and then the ones that are formed will divide, and they'll keep on doing that, and they'll keep doing it, and you can see now that a colony is forming each place there were bacteria. Eventually, then what you see is a colony, and then you count that colony, because now you can see them. Okay. All right, so that's why we call them colony forming units, and these are a good. The theory here is, and it's an important thing to think about, but the theory is, if you've got enough biocide, if you're doing things right in that tower, if you have enough stuff to kill Legionella, you should have enough stuff to kill normal bacteria too. So even though it's never correct to use a dip slide to tell you if you have enough, if you're adequately controlling for Legionella. The only thing that can do that is checking to see if you have Legionella. But it is sort of like the canary in the coal mine. It does tell you if you have a problem. And so we use these dip slides. They're pretty fast. You get results pretty quickly. And it tells us if we have a problem. And if you start seeing the number of So this illustrates the principles of a bacterial growth curve or exponential growth. Very simply, it just means that every time the bacterium divides, it makes 2 cells, and then 4, and then 8. You can see that in this little mathematical presentation. If we assume that the dividing time for Legionella is 2 hours, which is the most common number I've seen in the literature, although it certainly varies depending on the environmental conditions. But so you start off with 10 cells, and then 2 hours later you got 20, etc. 24 hours you've got 40,960, and 48 hours you got 168 million cells. And if you graph that out, you can see that once the thing gets going, it doesn't take very long until you've got a real problem on your hands. Imagine that many bacteria in the water as the tower is evaporating and the water droplets are being sprayed into the air. This can be a real problem. And this is how people end up getting sick. They inhale these droplets with all these bacteria on them. Bacteria is this. So both of these are Legionella growth incubators. They're different though. And so how do these incubators, these Legionella incubators, differ? Obviously one of them is much bigger than the other one, but there's another difference that really gets to why we have to monitor these things so closely. If you're lucky enough to have a hot tub at your house, or a spa, then you know that you can add chlorine or bromine or a lot of these chemicals to it that keep that level at a constant that's sufficient to inhibit the growth of harmful bacteria. And you go in and you stick a little dip thing in there, a little paper like pH paper or something, and it'll tell you if there's enough chlorine in there very quick. If not, you just add a little bit and you're done. Okay. Not so simple with these big honkers like this one here. And why is that? Well, here you're taking the same water and you're recirculating it. So if you add chlorine to it, until that chlorine reacts, it's going to keep circulating throughout your system and it's going to keep controlling the bacteria. But these, because of the constant evaporation, we have to keep adding fresh water. Not only to compensate for the evaporation, but also because salts start to build up, right? You get scale and all that. So, and when you're adding fresh water, what happens is you got to take something out too, right? So, if you're taking water out that contains biocide, when you add it, you have to make up for that biocide. So we are constantly diluting the biocide in these cooling towers. And so we always have to keep adding fresh biocide to it. If not, what we'll end up with is no biocide, or levels that are so low that Legionella will start having breakfast on us. So that's why we really have to pay close attention to these towers. And that's, the part that you're taking out, as you know, is the blowdown. So we have to keep on replacing that. All right. So another thing you need to do is you need to prevent stagnation. Why is that? Well, when the water doesn't flow well, the resulting areas that don't keep flowing, they encourage the growth of biofilm for a lot of reasons. One of them is that if you're not constantly getting fresh water into that area, fresh from the source, then that water doesn't contain, it kind of stagnates. The biocide that's in there can react and you lose the amount of active biocide. That lets the Legionella start growing. So the other thing is, of course, that the temperatures can vary significantly if you're not constantly circulating water. So one of the principles in maintaining towers is you need to have good circulation so that the chemicals and the temperature and all that stuff are adequately controlled. And because if you do that, then you should be able to control Legionella. So the other thing we mentioned earlier was that we need to maintain the equipment properly. You're only as good as what you're working with. And if your equipment's not working right, then it's going to be really hard to control Legionella. So all those principles we mentioned earlier, we are trying to prevent biofilms from forming. If the equipment's working properly, you can keep organic debris from building up. And organic debris can be anything from yeast to just byproducts of growth, and chemicals that are corroding the facilities and the metals, scale building up, all this stuff. And so all of this goes together to make really a nice environment for the growth of Legionella, and that is what we're working so hard to try and stop. So it's important to make sure that not only are we doing what we're supposed to do to keep the biocide and all that stuff going, but that we also are maintaining the equipment. That's why we have industrial cleaning done a couple times a year. That's why we spend so much time trying to make sure that we control all of those things that can lead to scale and damage to the equipment. So I know I probably haven't answered all your questions, and I wish I could be there in person to do so. But here's how you can get a hold of me. So I'm pretty sure the guys you work with, a lot of them, certainly, those folks that have been around for the last couple of years working on this can answer a lot of your questions. But, don't hesitate to write to me. I'll do my best to answer your questions. Just send it to this email address. As I'm sure you probably know, a lot of us are working off campus right now, but don't hesitate to contact me, okay? All right. Thank you all very much, and be safe. Creep up. It means somewhere something isn't right, because theoretically these things should not be able to grow to very high counts. So that's why we do that. All right. Now, one of the things I like to show people is